Hello there, welcome to this special issue of Reproduction, which is celebrating the 25th anniversary of the birth of Dolly the sheep and, with her, the emergence of what we know today as cloning by somatic-cell nuclear transfer. My name is Kevin Sinclair. I am a Professor of Developmental Biology at the University of Nottingham here in the UK. My association with Dolly is indirect. For a 20-year period between 1993 and 2013, I either collaborated with or worked alongside the two pioneers of this scientific achievement, that is, Professor Sir Ian Wilmot and Professor Keith Campbell. My own interests concern the development and refinement of advanced reproductive technologies, leading to the birth of viable and healthy offspring. It's testament to the significance of the achievement of Wilmot and Campbell that some 25 years later, we are celebrating not the birth of a human, but that of a sheep. The birth of Dolly represented a seminal moment in the field of developmental genetics for several reasons. It first provided clear evidence of genomic equivalence between embryonic and somatic cells, demonstrating that it is possible to re-establish a pluripotent state in differentiated cells. In so doing, it inspired Shinya Yamanaka to later identify at least some of the factors required to induce pluripotency in cultured somatic cells. This led to the creation of so-called induced pluripotent stem cells. Yamanaka would go on to share the Nobel Prize in Physiology or Medicine with Sir John Gurdon in 2012. The achievements of Wilmot and Campbell, however, did not go unrecognised. There were many accolades over the years, including the 2008 Shaw Prize in Life Science and Medicine, often considered to be Asia's equivalent to the Nobel Prize. This they shared with Dr. Yamanaka. In this anniversary issue of Reproduction, we present 7 review articles from leading scientists in the field, including those at the forefront of the technology 25 years ago. This issue opens with a brief historical overview of cloning by somatic-cell nuclear transfer, followed by articles that discuss how this technology has developed during the last 25 years. Scientific advances in nuclear reprogramming and underlying epigenetic mechanisms are then discussed. This is followed by consideration of future applications of cloning in the context of animal breeding and the generation of genetically modified animals with agricultural and biomedical applications in mind. Prospective stem cell-based approaches are then discussed. These may yet emerge to become the most effective means by which to establish genetically modified animals in future. Finally, a number of ethical concerns initially raised following the birth of Dolly persist to this day. These are considered in the final article of this issue in the context of technologies that influence genetic inheritance in both humans and farm animals. It would seem that 25 years after the birth of Dolly, the debate around the safe and ethical use of these advanced reproductive technologies is still going strong. I hope that you enjoy this issue, and I'd like to thank the editorial board of Reproduction and the production team at Bioscientifica for supporting me and all the authors in producing this anniversary issue. Thank you very much, and I hope you enjoy the issue.